Okay, in this demonstration video we're going to demonstrate for you the InstAlert 10-panel drug testing kit. Now this is one of the first drug multi-panels to be introduced into the UK marketplace. This InstAlert branding is a professional CE marked product sold in cartons of 25 individually foil wrapped test kits, and what you're looking at here is one of those foils. InstAlert is the trademarked brand. Innovacon is the manufacturer based in North America. CE marked, and on the reverse of the foil you'll see the basic coding information, which is the multi-drug one-step test panel screen DOA 1104. Standard coding for a 10 panel is 1104 with the InstAlert product, and you'll also see the drug abbreviation codes, which is the standard format that's chosen on that 1104 panel. This is available in a multiple choice of different panel combinations, but our standard 1104 in the UK is cocaine, amphetamine, methamphetamine, cannabis, methadone, ecstasy, opiates, barbiturates, benzos and TCA. Now also on the reverse of the foil you'll have the lot number and the expiry date. It's marked as an in vitro diagnostic for human testing purposes. This is a medical certified in vitro diagnostic device, so it's got IVD certification, that's part of its quality certification. And the storage on the membranes in foil should be between 2 and 30 degrees. So if we show you what you get when you actually open up the foil and remove the card, this is a standard format for multi-panel InstAlert drug screens. You've got example positive and negative results showing there, an area for ID of the sample, the dates it's taken from, and the operator's identification if required. Now in these windows here you've got the coding for the individual drug groups, 5 on each side on a 10 panel. So if we turn it over you'll see almost identical information on here with a different set of test panels. And to conduct a test, very simple itself. Remove the foil cover, and there you have, if I show you this end on, in this case a set of 10 prongs coming out the base with individual wavy lines. The maximum dip line on a 10 panel and any other multi-panel is the actual base of the plastic holding, so you don't want to dip it further than that. If I just take this panel now and apply it into a sample, you need to apply this into a sample for between 3 and 5 seconds. If I bring that back into the view of the camera now, you will see travelling up in the test window area, you'll see the pinkish dye travelling now osmotically up from the prongs where it's been absorbed. And at this stage it's perfectly okay for you to recover the prongs. And we're basically now waiting for the development of control and test lines. Now the beauty of the multi-panel test is that you've got clearly marked on these panels the control line area and the test line area. So it's very easy to interpret. You're looking at individual drug codings on here, MDMA for ecstasy, MOP for opiates, BAR for barbiturates, so it's almost self-explanatory. And you can see on this all negative test result that even now at 1.5 to 2 minutes after applying the sample to it, you can interpret the results on this panel as all negative. You've got control lines showing in every window and you've got test lines showing in every window. I think it's quite useful to be able to show you the variance that you get on some of these test lines. If we have a look at the TCA line here, which is the one furthest to the right of your screen, you'll see that the test line showing on the TCA membrane is very much fainter than the corresponding BZO test line in the panel window next to it. Now that is of no consequence whatsoever in terms of the interpretation of your results. You have a discernible test line there showing, and as long as you have that at the 10-minute cutoff, you must interpret that as a clear negative result. I've got a faint test line showing at 1 to 2 minutes, so I'm interpreting that as a clear negative test result for TCA. Even though it's much fainter, thinner, and not as colour dense as the BZO, you must not try and draw any inference by that. It is a negative test result. So at that stage, after 1.5 to 2 minutes, if you've got all negative results on your panel, that's it. Your test's complete. You can move on to the next test that you have to perform. So you are looking at a turnaround and operating time in terms of negative results being shown on these test panels in 1.5 to 2 minutes. So it's quite physically possible to be able to do, one every 5 minutes in terms of a screening procedure. That's the sort of time you should be allowing yourself. The problem comes when you start getting positive results. Now, you have to interpret a positive result at the specified time that the manufacturer has given you on the full data sheet instruction that is supplied with these tests. And in the case of InstAlert, that's 8 to 10 minutes. So if you have no test line showing at the 10-minute stage, then that is a positive result. But obviously that does slow your testing procedure down somewhat, that you've got to give it that period of time. So the only additional piece of equipment that you're going to require to conduct these tests really is some form of timing device. That can be a simple wristwatch, stopwatch, whatever you have available. In this day and age it tends to be an iPhone timing system that you set going. They are extremely accurate and reliable. You're looking at, across the 10 range of membranes here, between 97 and 99% accuracy. Each specific membrane has been batch certified and will have a certificate available on the batch and lot number should you require that. These are non-evidential tests in terms of how you can proceed with the information. They are screening tools intended for wheeling out all your negative results quickly, easily and cheaply. As soon as you hit a positive test result on one of these panels, you will be required, if you are going to take any action or move to an evidential arena, to confirm the test results using an accredited laboratory service. Now that means basically either containing your sample or taking a further sample and sending it to an accredited lab who will conduct a GCMS test, that's gas chromatography mass spectrometry, which is still considered the gold standard in terms of testing in this country, and they will provide an evidential certificate confirming or in some cases showing that your positive result was not down to illicit drugs but may be down to substances like cocodamol or other codeine-based products that may give you a positive on the opiate test. Unfortunately, with these types of tests, you have no ability to determine whether a positive opiate test is down to cocodamols, DF118s, i.e. prescribed, legal or over-the-counter codeine and opiate-based analgesic products. You cannot differentiate those on a simple dip test from street heroin or opiate use without proceeding to a GCMS.